Namaste to all students. Khushbu here. Myself JV, Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about Plasmid, and this session is related to recombinant DNA technology course of program BTech FBT, BTech FBT 4th. So first of all, what do you understand by the Plasmid? And Plasmid vector is basically very much important in terms of molecular cloning, and the basic example of the Plasmid vector is P blue square. So Plasmid basically phase from M13 bacteriophage and mid from plasmid. It contains 1500 base pair. If we talk about the construction of plasmid, so it is having origin of replication, leg Z region. Leg Z is region is basically for the identification of selection of the recombinant vector. So it's basically identification factor. And third one is the multiple cloning site. MCS, multiple cloning site, is basically having more than 2 to 5 restriction enzyme site, cleavage site, so that the target DNA can be introduced into the multiple cloning site. And fourth one is the origin of replication, that reason from where replication will start of vector or recombinant vector. And fifth one is the ampicillin resistant gene. This is also a selection marker for the recombinant vectors. And if we talk about the use of plasmid, so plasmid is basically used as cloning vectors, sequences in vectors, and expression vectors. What are the advantages of the plasmid? So it can be used to provide single or double stranded material without any reclone. The carrying capacity of the plasmid is higher than the phase vectors, and plasmid has higher efficiency in transformation process than phase vectors. Plasmids are genetically more stable than recombinant phase vectors. Plasmids can be exploited to generate single-strand DNA template for the sequencing purposes, and single-strand plasmid vectors inside the phase can be targeted by or targeted for site-directed mutagenesis. If we talk about the P-blue script, which is the example of the plasmid vector, so P-blue script is an example of a combination between plasmid and phases, which they create the plasmids. Plasmids represent a hybrid type of the class of the vector that serves to produce single-stranded DNA. What are the features of the P-blue script or plasmid? So first feature is like puck vectors, which P-blue script is derived from. There is a multiple cloning site inserted into the leg Z gene. Second one is the P-blue script also contains the origin of replication of the single strand phase F1, which is related to M13 phase vectors. This translates into that a cell harboring a recombinant plasmid, if infected by F1 helper phase that supplies the single strand phase DNA replication components, it will produce and package single stranded plasmid DNA. Now we can understand more easily about the P-blue script vector structure by its diagrammatic representation. So you can simply see P-blue script second SA plus or minus plasmid. Here you can see this is the origin of replication, from where single stranded DNA can be synthesized, and this is ampicillin resistant gene. This is antibiotic marker for the selection of the recombinant vectors. This region is having puck vector origin replication, and this is multiple cloning site MCS, which is basically present in the leg Z region, and it is having SET1, KPN1. These are restriction sites, and the length of the P-blue script second SK is approximately 3 kilobase pairs. Third point is basically produce RNA transcripts. So PBS has a T3 promoter on one end side and T7 promoter on the other terminal end. This is key because it enables one to isolate the double-stranded plasmid DNA and transcribe it in vitro with either of these two-phase polymerase to produce pure RNA transcripts to coincide with either of the two different strands. If we talk about the summary of the P blue script, basically it is having 2091 base pair length. It derived by replacing PUC19 polylinker of PBS plus minus with synthetic polylinkers. SK designation indicates that polylinker is oriented such that the leg Z transcription proceeds from SET1 to KPN1. 21 unique restriction sites in the multiple cloning region basically present in the P blue script, and blue-white color selection also present due to the leg Z region presence in cloning vector. Uh, there is a basic concept for the leg Z region. If leg Z region is active, means there is no any kind of a MCS region, or like there is no any kind of a incorporation of foreign DNA. So leg Z region will be active, and it will generate the beta galactosidase enzyme, and in the media where we grow the recombinant E. coli or normal E. coli cells, they have to be mixed with the lactose and dicot. When normal cells of E. coli, which is having normal uh, plasmid vector, so they will react with the media of nutrient media, and they will convert the dye into blue color because of the presence of beta galactosidase. It will break down the dye and will create the blue color. So that's why we can confirm that E. coli cell is having this plasmid vector, which is non-recombinant, because Laxer region is active, because there is no any insertion of foreign DNA or target DNA into the MCS region of Laxer region. On the other hand, if E. coli cell is having vector, plasmid vector, but recombinant vector, means target DNA or foreign DNA has been introduced into the multiple cloning site MCS of leg Z region. It makes the leg Z region inactive because there is a manipulation in MCS region. We have introduced the uh, foreign DNA or target DNA into the MCS region. So that's why that MCS region is now changed, and this results basically leg Z inactivation. After the inactivation of leg Z region, that vector is not capable to synthesize beta galactosidase enzyme. So overall, that recombinant E. coli cell is also not able to synthesize beta galactosidase enzyme. When this recombinant cell grows into the media, which is having lactose along with the dye, so there is no beta galactosidase, so that cell will not degrade, or that cell will not break down the dye and will not produce any color. So cells will be simply creamish in the color or white in color. So that's how you can differentiate the recombinant E. coli cell and non-recombinant E. coli cells. Means recombinant cells will not able to break down the dye to produce the blue color. On the other hand, non-recombinant cells are having vector but not recombinant vector. And in that case, leg Z region is active because there is no manipulation in the leg Z region, MCS region, and that leg Z region will produce a beta galactosidase. This beta galactosidase have the ability to break down the dye with lactose and will produce the blue color. So non-recombinant cells will show the blue color. On the other hand, recombinant cells will show the white or greenish color. So that's how you can recognize the pre-blue script, recombinant and non-recombinant. Next one is the in vitro RNA transcription with T3 or T7 RNA polymerase. So these are also characteristics of the P-blue script. Next one is the double and single stranded sequencing. So both kind of a strand sequencing present in the P-blue script or Plasmid vector. P-blue script SK either the plus or minus orientation. What do you understand by the plus or minus orientation? So the plus and minus denote which single strand of the DNA, either the sense or antisense strand, can be rescued using a helper phase. These plus or minus orientations occurs in the F1 region of the plasmid. Thus, F1 plus origin consists of 3 to 4, 5, 9 base pairs. F1 filamentous phase origin of the replication that allows for the recovery of sense strand of the leg Z gene. On the other hand, F1 minus origin consists of 3-2 459 base pairs. F1 filamentous phase origin of replication allowing recovery of the antisense strand. So that is the basic difference in between the F1 plus origin and F1 minus origin. So this is how this session is sum up now. And if we talk about the key points and important points or important questions related to the plasmid vector. So first of all, what is the chemical composition and how it is prepared. So you have to know that plasmid is basically combination between the plasmid and phases. Generally, puck vectors are involved as a plasmid, and phases vectors are involved, like M13 phases are involved as a phases. Both characteristics of the plasmid and phases comes into the plasmid vector. And the best example of the plasmid is P-blue script. And if we talk about the component of the P-blue script, so you have to learn about the constructions of the plasmid, which is having origin of replication, like that reason for the selection of recombinant and non-recombinant cells, and multiple cloning site for the incorporation of the target sequence of DNA or RNA, and origin of replication, ampicillin resistant gene also required for the selection of recombinant and non-recombinant vectors. So these are the key points which are very useful for examination point of view. And this is now this session is sum up, and this session is powered by digital version 2.0 JAYOTI VIDYAPEETH University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.